Now that you have a good understanding of recombination and dihybrid analysis, it's time to extend some of these ideas to a new phenomenon that we haven't encountered before. And this is the issue of epistasis. What happens when we have two loci that have competing phenotypes that are not compatible with one another? So for example, we could have one mutant line of, say, a plant or some other organism that has colors. And in this case, we have one mutant that's blue, we have another mutant that is white, and the wild type phenotype is purple. Now, I've modeled this as two recessives, where each one is recessive to its corresponding wild type allele. So the white line has the wild type allele of the B locus, and the blue line has the wild type allele of the A locus. And this is why we see the wild type in the F1, but more importantly, we also see wild type in the F2. Now, if these two loci are unlinked genetically, then they will produce a 9:3 to 3:1 ratio when this dihybrid is selfed or intercrossed, depending if it's an organism that can be selfed or not. So the fact that we see wild type in the F2 when we cross two mutant lines together, as we've emphasized all along, this indicates that we're dealing with two different loci, and thus they need to be assigned and defined as different letters, as we've done already. The 9:3 to 3:1 ratio is then, just like it has always been, as we've seen before, to give us the different genotype categories. Now, a 9:3 to 3:1 is a genotype ratio, and the fact that we're now about to discuss non-compatible phenotypes doesn't influence the fact that this will still be genetically a 9:3 to 3:1 ratio. The question really is, what happens with the double mutant genotype? What phenotype will it have? Presumably, it can't be both simultaneously blue and white. Those are non-compatible phenotypes. The only way to determine what phenotype it will actually have is to look at the data and to see what phenotype it has. That's not something that you can predict in advance. If, for example, we said that this has a white phenotype, simultaneously, when it's mutant for both loci, it has the white phenotype that we associate with the little a genotype. The issue is we've got two non-compatible phenotypes. Does the blue genotype win and exert its phenotype and mask over the white? Or does the little a genotype with its white phenotype mask the blue? And in this case, because we look at the data and we see that this category is white, we would say that this genotype is masking the effects of this genotype. Or what we would say is the A locus is epistatic to the B locus. Epistasis just comes from two Greek words. Stasis means standing, and epi means upon. So what we're saying is that the little a genotype in this case stands upon the little b genotype and exerts its phenotype as if it was a contest between the two, and the little a genotype won. The way to draw this as a shorthand symbolism would be as follows. Here's the genotype that does the epistasis, and this curved line with a bar on it indicates that this genotype is exerting its phenotype and masking the phenotype at this locus. This is an example of what is called recessive epistasis, and the reason for that is that this genotype, the little a genotype, is a recessive genotype within its own locus. In other words, it needs to be the recessive little a genotype within the A locus before the epistatic effect can be exerted. The big A allele has no epistatic influence on the little b locus. It's only the recessive condition, little a, which does have that masking effect. So we would say that this is an example of recessive epistasis. So in this example where we've had the little a genotype epistatically exert its phenotype over the little b genotype, the net effect of that is to take this double mutant and group it with one of the threes. So specifically, now what we're saying is if little a is present, then it will mask the effect of little b. So the only necessity for having a white phenotype now is having little a. Doesn't matter if there's little b there or if there's a big B present or not. So the net effect of this is to produce a 9:3:4 ratio. So we have 9 that are wild type, or purple in this case. We have 3 that is the one mutant phenotype, the blue, which is allowed to express its phenotype when it's in the presence of a big A. And then we have 2 categories because of the presence of little a, where the white phenotype takes precedence even over a little b genotype. So that would come out as white. So this 9:3:4 ratio is an indication that we're dealing with recessive epistasis. Again, recessive referring to the actual genotype within the locus that's exerting the epistatic effect. Little a is the epistatic genotype, and the little a is recessive to its own corresponding wild-type allele, which means when this genotype exerts an epistatic effect, we call it recessive epistasis because this genotype here is recessive relative to the wild type at its own locus. So that 9:3:4, indicating that the 1 is grouping with one of the 3's, is a diagnostic for recessive epistasis. Let's extend this analysis now and include another mutant line, this red mutant. Same organism, but now just a new color. We previously discussed the white phenotype associated with the little a locus, and we've carried that forward. So here's what that cross would look like had we crossed it to a wild type. So we take a wild type pure line, a purple wild type pure line, and a new mutant red line, and we can see in this case the F1 is red, indicating that whatever is causing the redness is dominant to its corresponding wild type allele. We see a 3:1 in the F2, which indicates that this is a monohybrid. Now the only question is this an allele of the A locus, or are we dealing with an allele of another locus? The way to look at that, or the way to decide that rather, is to look at the F2 of the cross where you take the 2 mutants and cross them together. Now if I say that there are wild type, so there's purple organisms in the F2, this then indicates that this is a separate locus from the A locus, and we need to define it with another letter. So because the mutant phenotype is red and it's dominant, I'll give this a big R genotype. We already know that white is little a. I now need to define a wild type allele of the R locus. This obviously has a mutant allele of the R locus. The wild type allele I will define as R+, and I'll use a lowercase r to remind myself that the big R mutant is dominant to its corresponding wild type allele. The red mutant is only mutant in one locus. We see that from the 3:1 when we take this red mutant and cross it to the wild type line. So that means that this mutant line, being mutant in only one locus, also has a wild type allele of the A locus, which we had previously defined as a capital A. This gives us a dihybrid in the F1. And it's the recombination to a fully wild type genotype, or at least to a wild type to genotypes that give a wild type phenotype, which is what indicates to us that these are separate loci. If we only had mutant versions of R, for example, we would never have a wild type R in the F2, and we would never get purple. So the fact that we see a wild type phenotype in the F2 indicates that we're dealing with a dihybrid, and that therefore these 2 phenotypes are caused by mutations in separate loci. Okay, now it's time to take this F1 and self it, or allow it to interbreed depending on the organism. And now we'll look at the possible genotypes that can arise from this. So let's just sketch this out briefly. So again, assuming that these loci are going to be genetically unlinked, i.e. they are assorting independently on different chromosomes, or perhaps they're 50 map units apart on the same chromosome, genetically independent, we would see this same pattern, the 9:3:3:1 in the F2. The 9 would have both dominant alleles. One of the 3s has one of the recessives with the other dominant, and then converse, and then the 2 recessives together. And you'll notice that one of the wild-type genotype in this case, because one of the mutants is dominant, this is the category that has the wild-type phenotype. It's wild-type, has at least one copy of the big A, and we know that the mutant allele little a is recessive to the big A. So here we have a wild-type situation at the A locus, and we also have both wild-type alleles at the R locus. So phenotypically, this is the category that is going to give us wild-type, or in this case, purple. It would not be possible to get this type of situation where you have a wild-type phenotype if, in fact, we were dealing with mutant alleles of the same locus, as we've discussed previously. Now, the question is, what happens when you have both mutant situations at both loci? Is it possible to be simultaneously mutant and white and mutant and red? And in this case, because we're talking about non-compatible phenotypes with epistasis, let's imagine a case where you can only be one or the other. There's not sort of gradations in between. So here's the situation where we have both mutant situations present at the same time. And if I say that this is red, simply by examining the data, and again, you can't predict it in advance, you actually have to look at the data. If I say that this is red, then what I'm saying is that the R allele exerts its effect and masks the phenotype of a mutation at another locus. So in this case, we would say that the big R mutant allele is epistatic to the little a genotype. This is an example of what's called dominant epistasis, and the reason for that is because the allele that's doing the epistatic effect is itself dominant to its own corresponding wild-type allele. We've made the point before that dominance and recessiveness always applies strictly within a locus. So here, when we're talking about masking an effect at another locus, dominance or recessiveness is not the correct term to use. Epistasis is the correct term. If there's a masking effect going on between loci, it's epistasis, and then it's dominant epistasis if, in fact, the epistatic mutant allele is dominant to its own wild type, and it's a recessive epistatic situation, like we saw in the previous example, if it's a homozygous recessive genotype which is having that epistatic effect. So this would be an example of dominant epistasis.